little review of our genetics material. So let's get started. We're going to start with the vocabulary. If you're here to watch some of the other Punnett Square questions, you can scroll through the later part of the video and then find the part that you need. So we're going to start with the definitions. I'll just write down the definitions as we're going along. The first question says, what is a gene? So a gene is a sequence of DNA that codes for, I'm going to have to write for a protein or a trait. So what we mean by that is when we're looking at a strand of DNA, right? So DNA double helix, one DNA doesn't contain just one gene. There are a lot of genes on this DNA that gives you all different kinds of traits. Humans have 46 chromosomes, and if there's only one gene per chromosome, it doesn't really work out. So you want to think about a gene as an example. You can think about it in terms of a recipe and a recipe book. You can have 46 recipe books, but you're not just going to have 46 recipes. You're going to have a whole lot of recipes, but each recipe is in charge of making a specific dish, right? So similarly, a specific part of your gene, let's say this is gene 1, let's say this is gene 2, the different genes will code for different traits. Gene 1 might be coding for eye color, whereas gene 2 might be for hair color, okay? Moving on to the second question. Well, a gene, obviously. All right, what is a genome? A genome is all of the genes and all of the chromosomes. So you can say all the genes on all the chromosomes, say in an organism. Okay, so we're saying if we're talking about your genome, it will be all the DNA that your body carries. That's what we mean by a genome. All right, moving on to the next question. What is a locus for number 4? So like I said earlier, we have these genes that are located on one particular chromosome. So that's one chromosome, right? In our bodies, we have homologous chromosomes. So instead of just this one chromosome, you will really have another chromosome that can pair up with it during meiosis. So why would you have these pairs of chromosomes, right? Why do we say 23 pairs of chromosomes and not just 23 chromosomes or 46 chromosomes? It's because the 23 pairs of chromosomes are really 23 pairs of what we call homologous chromosomes. So these homologous chromosomes are passed down from mom and dad. You receive one chromosome from mom, and then you have a very similar chromosome passed down from mom. The majority of the DNA on those two chromosomes are exactly the same, but parts of it will be different, which is what makes mom different from dad and what makes you different from either your parents, right? You're a mix of everybody's DNA. So here's the deal. When we have gene 1 over here on the homologous chromosome, on the other DNA, you also have gene 1. So if this were to be a hair color gene, dad's chromosome, this one is also a hair color gene. So they're coding for the same trait. You would say gene 1, the hair color gene, together, even though we're looking at 2 of them, it's still one gene. It's like, even if I had 2 pens, right? I'm still looking at one category of material, pen, right? If I'm looking at 2 phones, then I'm still looking at one category of object, phones, right? But if I'm holding 2 pens and 2 phones, how many different types of objects do we have? We got the phones and we got the pens, right? But I can have 2 different varieties of the phones, 2 different varieties of the phones, and 2 different varieties of the pens. Okay. So what am I trying to say with all this extra information is that you will have the same genes located at the same locus. So you would also have gene 2 over here. Gene 1, my pointer. Gene 1 over here and gene 1 over here are at the same exact locus on the pair of homologous chromosomes. So the one from mom, one from dad, for the hair color gene, they're all located at the same locus, okay? So you can say for locus, the location of a gene on a chromosome. Okay. Next question. Number 5, it says, what is an allele of a gene? An allele of a gene is, you can say, a version of a gene. So when we say dominant versus recessive, you can have dominant versus recessive trait, and you can have dominant versus recessive allele, right? So what do we mean by dominant recessive allele? That's your big A or little a. There are two different varieties of the DNA. You can have the DNA, or part of the DNA, you can have a gene, right? Like gene 1 over here might be coding for brown hair color, whereas this hair color of the other DNA might be blonde hair color, right? But they're both for the trait hair color. So it's still the hair color gene, even though you can have two different varieties of it, okay? So we say an allele is a certain version of the gene. You can have the dominant allele versus the recessive allele. All right, moving on to the next question. Number 6, how many alleles of the same gene does a parent carry? So when we're looking at a parent, you have your 23 pairs of chromosomes. You do have homologous chromosome, right? So here's your homologous chromosome. If you have a gene right here, you would have another gene right here. If you have a gene right here, you will have the same gene right here. But these two are the same one. You might have big A over here, little a over here. Maybe you have big B and another big B, right? But how many alleles of the same gene? So for A, there are only two A's, right? It's like the two pens. So there are only two varieties of the same gene in a somatic cell. Okay, so your 23 pairs of chromosomes, you have double of everything. You get, like as a parent, you also have half of your genes from mom and half of your genes from dad. So you have two of everything, okay? That's why when we write out the parent's genotype, it's always like big A, little a, or little a, little a. All right, question number 7, how many alleles of the same gene does a gamete carry? So when we're looking at a gamete, there's only one allele being passed on, right? That's why when we're looking at big A and little a, only the big A gets passed down to the child, or only the little a gets passed down to the child. You're not going to get both from mom, right? Mom will just give you this one or that one so that you get your other A from dad, right? Mom gives you one copy of, for example, the hair color gene, and then where do you get your other one? You get it from dad. So it's so important. It's very important for your mom to only pass down one copy of the gene to you so that you get another copy from the other parent. Okay, now, how many alleles of the same gene? So once again, we're talking about like how many copies of the genes do you get? You receive 2 copies. One is from mom and one is from dad as a child, right? Offspring is the same thing as a child. Maybe you wouldn't call certain babies like a bacteria offspring, you wouldn't call it a child, but it's okay, you got it. All right, what is a dominant allele? A dominant allele, you can think about it as big A, but what does that mean? Dominant allele masks or like represses the recessive allele. Okay, and it always so you can say the trait is expressed with 1 or 2 copies of the allele. So what am I talking about over here, right? That's a lot of words, but really it's important for this one. You should really understand, like, what am I trying to say over here? What I'm trying to say is that when you have a dominant allele, right, the big A on its own is not a trait. That's just your gamete. But if you had big A, right, you can have 2 of them at the same time, and you will have the dominant trait. So let's say big A is brown hair. If you have 2 copies, you'll still be brown hair, right? If you get a brown hair gene from mom and a brown hair allele of the hair color gene from dad, you obviously will have brown hair. However, if you have a brown hair allele from mom, and let's say blonde hair is little a, and you get that gene from dad, you will still be showing the big A's trait. The little a is like suppressed. It's as if it's not there, but you still need the two copies. You just wouldn't see it, right? It's like mixing paint color. If you're mixing brown and like sky blue, right? Chances are you're going to see the brown color a lot more than the sky blue, even if you put in a lot of blue, right? Dominant allele is powerful like that, but it doesn't mean that it shows up the most often. Just because it's a dominant allele doesn't mean it's the most common that you see in a population, okay? So the recessive allele, you can say, only expresses when there are two copies. Or you can say, you can remember it gets like repressed by the, I'm writing so slow, dominant allele. Okay, so what I'm saying over here, in order for the little a to show, like the DNA is obviously there, but the trait that the little a is coding for, it will only show it if you get two copies of it, right? If you get one dominant allele with it, then you're not gonna see the little a, right? The little a you're not gonna see, you will be seeing the big A, even though they're both there. Okay. So, oh, whoops. That's what we mean by only expresses when you have two copies. All right, homozygous. Ooh, we're on to the easy stuff. Not as much writing. Homozygous when you have two of the same alleles. We're just not following the grammar rules as long as you understand it. Okay, so you can have big A, or little a. Maybe you can have big A, big B. But either way, you have the same alleles for the gene, right? Like if you have big A, big B, and little b, you're not going to say it's homozygous for the B gene. That's a different gene. Okay. All right. Next question. We have heterozygous, two different alleles for a genotype. So this will be your big A, little a, right? One dominant, one recessive. A genotype is the gene an individual carries. Okay, so how do you know if something is a genotype? You're looking at the letters. You're looking at the big A, little a, little a. Those are all genotypes because we're not looking at the actual physical trait, right? Well, let me plug it in. When we're just looking at the big A, little a, right? All of these, it doesn't give you the actual trait. You don't know what eye color, hair color it is. It's just like, what kind of DNA do you have? Do you have the dominant? Do you have the recessive? How many do you have those? All right, next one. Phenotype is the physical trait, obviously. Okay, so that's what you actually see, right? When I look at you, I don't see your big A, little a. I can look at you and take a guess, right? If you have the recessive trait, the outward physical trait, then I can say, oh, you're probably little a. But what I actually see is the phenotype, like the actual physical trait, your eye color itself, not like the DNA. Okay. Next one, monohybrid cross is when you're looking at one gene. Okay. So that means you're just looking at the big A / the little a. Like you're just looking at the hair color gene on its own, right? You're not mixing up the A and the B's. It's just one type of letter. A dihybrid cross has two genes. Also, for the monohybrid cross, with one gene, you can have two different alleles in an individual. Right? So what are we saying over here? That's when you have big A, little a, little a. You need two alleles to make one person, right? Or whatever animal. Two genes, that means you have four alleles, right? For example, you can look at big A, big B, little b, right? A is for maybe eye color gene. B is for the hair color gene. So now these are two different genes, like two types of instructions that will give you the actual physical trait of eye color or hair color. But because you need two copies for the eye color and two copies for the hair color, together you get 4 alleles in total. All right, now we look at the generations. P is parental generation. That's the parent, like what's on the outside of the Punnett square. The F1 is the first offspring generation. So that's what's inside your box. But if I were to say, like, we have plants, we get the next generation of flowers, and then we cross those flowers and get another generation of flowers, that's your F2 generation. So second offspring generation. All right, moving on. These questions are taking a while. I just write slow. Number 20, what do the squares represent in a Punnett square? So when you're looking at a Punnett square, it's really like these 4 squares, right? You can put letters on the outside, for example, but it's asking about those squares on the inside. So they represent the offspring, right on the other side, genotype. Okay, so the squares themselves only represent the genotypes, but based on the genotypes that you use, that you predict, then you can use that genotype to then figure out, like, what does this genotype represent in terms of the actual physical trait? What physical characteristic would it be? Okay. Number 21, what do the letters outside the Punnett square represent? So now this one, we're talking about like the letters on the outside, the individual letters. Those letters represent gametes from each parent, right? Like I said, each parent carries 2 gametes. It's as if I had 2 pens, one of them is a black pen, it looks pink. Let's just say it's pink. One of them is a pink pen, the other one is a orange pen, right? I have 2 pens, but I'm only going to give you one of the pens, right? So you can end up with the orange pen, or you can end up with the pink pen. Now, your parent, like I said earlier, has 2 copies of each gene, right? Mom, for example, has either big A or little a, but she's not going to give you both of them. She will give you either the big A or the little a. So those, like, the reason why we separate the 2 letters is to represent that you will get this big A from mom, right? And then you will get this other big A from dad. You end up with 2 big A's, not because you got both of them from one parent, you got one from each parent. So on the outside, once again, these are gametes. Okay, just make sure you understand it conceptually. What are we looking at? All right. Oh, the next one. We have the purpose, or on the next page, which change color. Test cross. The purpose of the test cross is to figure out, how do we say it in better English? Identify the genotype of a parent with the dominant trait, or you can also call that the dominant phenotype. Okay, so what are we talking about over here? So when you're looking at a parent that has a dominant trait, let's say we're doing our big A versus little a. Let's say big A is brown eyes, right? Little a is blue eyes. When I see someone with blue eyes, well, little a is the DNA that will give you blue eyes, but little a on its own is not going to give you blue eyes. You need 2 of them, right? And why would you have 2 little a's? Because little a is recessive. The only way that you can see the recessive trait of the blue eye trait, right? These are on the left, the one letter is the brown eye allele and the blue eye allele. To have the blue eye trait, you need 2 little a's. Now, if I have someone with a brown eye trait, I don't know what it is. The brown eye trait could be big A, or it could be big A, little a. I really don't know which one it is. So then I'll have to perform something called a test cross, okay? So how do we do a test cross? For a test cross, number 23, you, excuse me, you will take, excuse me, you will take a dominant, like an individual with a dominant trait and cross it with a homozygous recessive, right? Well, if you know it, then it's right. But if you don't, Now you're learning it. Okay, so what do I mean by that? If I'm looking at this guy from earlier, guy, girl, doesn't matter, with a brown eye trait, right? I don't know whether it's the big A or big A little a. All I know is that it has brown eyes, right? As an example. So what I would do is I would take the brown eyes, I will cross it with blue eyes. Let's say these are dogs, okay? It's a little creepy if it was people. Take a brown eye dogs and cross it with the blue eye dogs, right? Because the blue eyes, I know, are little a, little a. So then what do? Well, I'm trying to figure out if this one is big A, little a, or big A, big A. I don't know which one it is. So after I cross it, I look at the children, okay? So let's try Punnett square. If my unknown is big B, and I cross it purposely with big B, little b, right? You can very quickly find out. You don't even have to fill out all 4 boxes. You can see that you have big B, little b. So that means all the offspring will have brown eyes. Okay? So what does that mean? If all the offspring, okay, so if all the offspring have brown eyes, that means going backward, the parent, oh, that's not a highlighter, the parent that's unknown is really big B, big B. However, if I were to do a cross, and then I find out some of the children have brown eyes, some of the children have blue eyes, let's see what happens, right? So this is a hypothesis, like a hypothetical Punnett square, right? So we're looking at the Punnett square, and I see in this particular scenario, in this type of cross, like if the parent was this, what would happen in the child? You would have half brown and half blue, right? These two are the blue, okay? So if I look at the children, and the children has half brown eyes, half blue eyes, what that tells me is that the parent that was unknown was a big B, little b situation. That's heterozygous, okay? All right. We'll do some practice problems. Let's look 2024. Dihybrid cross, how many traits do we look at during a dihybrid cross? We look at 2 traits. So instead of just looking at big A for little a, for brown eye, blue eyes, we also look at big B and the little b's, or maybe it's big B, I don't know, right? But there'll be 4 letters that you look at. You have the eye color gene, and you also look at the hair color gene. So then you're looking at 2 different physical traits. How do you find out the gamete? You use the FOIL method. It's 8:24. I'm going a little crazy. Okay, so how do we do the FOIL method? As an example, let's say we have big A, little a, big B, little b. Move it closer. This is the parent genotype. So what would I do? I will FOIL it. What I mean by that, I have 2 traits. I want to think about the A trait together and the B trait together. So F for first. So we look at the first of each type of letter. Okay, so the first of the A's and the first of the B's. That will give me big A, big B. So I'm asking you on the test to do FOIL, just follow the same order. Okay. O for outside. We're looking at the first and the last of the letter, big A, little b. And then we have I for inside. The inside 2 letters, we have little a and big B. And then we have L for last, little a, little b. So L for last is saying the last of each group of letters. So the last of the A and the last of the B gives you little a, little b. So in this case, we have 4 different gametes. But let's say I'm looking at big A, big B, big B. You can FOIL it, right? If I tell you to FOIL it, right? First, outside, inside, last. Well, you quickly find out the only type of gamete that you have is big A, big B, because you only have big A, right? You only have big B. There's no way that you can get a little a out of this situation. So if I'm asking you to write out 4 gametes, then you'll write big A, big B 4 times, right? That's the quick way to figure out your gametes. Let's try another one. Little a, big B, big B. Just by looking at it, do you know the gamete without foiling? Well, you should, because the gamete is little a, big B, right? I only have little a, and I only have big B. So that means the only trait I can pass down is little a. Wait, so not the trait. The alleles, the only alleles I can pass down is little a and big B together. Okay? I wouldn't just pass out little a. If I'm looking at two traits, I'm looking at two traits. Two letters, two different letters. If you write little and little a, obviously I can't punch you, but I'm not, no physical harm, not threatening at all, okay? But I'd be a little disappointed because there's A gene and there's also B gene. You got to get both, right? You can't just get the hair color gene from your mom and not the eye color gene. That would be very troublesome. That would not be good, right? So if I'm looking at eye color and hair color, you need both. You got to make sure you get both, okay? Whew. Now, let's look at some practice. How long is this video gonna last? I don't know, but may Lord have mercy on all of us. Okay, let's start with the monohybrid practice. Easy. Okay, so when you're looking at a monohybrid practice, you're just looking at one trait. So let's look at the questions, answer the questions, just speed around. You want to do this as fast as possible, so that when you're taking the test next Tuesday, you're not wasting time trying to figure out, like, how do I do this again? You just do it, okay? Just do it. All right. For this question, it says, in dogs, a dominant gene, W. So it gave you the W, use the W. But if you're writing W, make sure your big W and little w are very different from each other. Okay, I think I can switch to it. Okay, it says, big W gives you wire hair texture. Little w, smooth hair, okay? Notice the good practice that I'm doing, okay? As a teacher, I am still underlining and doing squiggly lines. You should do the same. Well, why is that? It's hard to look at a whole paragraph, right? Or if you're answering a question, you might miss something. But if you're drawing your eyes, this is such a side note. If you're drawing your eyes to the most important information, it's much easier to get the questions right because you're wiring your brain to pay attention to the wiring. You're wiring your brain to pay attention to the important information, so you're not going to get it backward by accident, okay? Good practice, do it. I'm telling you, do it. All right. So it says, a homozygous wire-haired. Now, homozygous means two of the same. Wire-haired, as I underline, is big W, okay? So that's homozygous. It says male. It's telling me that it's a male. It really doesn't matter. The male and the female for a regular monohybrid, they're all written the same way. Okay. Then we have a female. I'm circling it just to point out that it's a female. And once again, it doesn't matter. Okay. In this case, it doesn't matter, but if it's excellent, it does matter. Okay. Wire hair to a smooth hair. Now, well, you're like, is this homozygous? Is it heterozygous, dominant, recessive? Well, here's the deal. Smooth hair is little w. The only way, oh, sorry. And also little w is recessive. The only way that you can have a recessive trait shown is when you have two of them, okay? So that's when you have little w and little w for mom. I'm going to use a different color. This is mom, okay? Now, then you do the Punnett square. And as a reminder, this is a gamete from dad, another gamete from dad. They're the same, but it's okay. Gamete from mom and gamete from mom. Okay, so how do we fill out a Punnett square? We get a big W from dad, little w from mom. I don't know why I just did that. Okay. Same thing. Okay, well, this is easy, right? We have the genotype. We're looking at the offspring. So we're looking at these four boxes. As you can see, they all show the dominant trait because they're all heterozygous. I'm gonna write heterozygous. And what happens when you're heterozygous? You show the dominant trait. And the dominant trait in this case is wire-haired texture. So the genotype is what you see directly in the Punnett square. So big W, little w. It says ratio. So we're going to be good and we're going to do the ratio. Okay. We can do big W to big W, little w to little w. Whoa. Little w equals to 0. You can do 4, 0. Or maybe you can do 0 and 1 and 0. Okay. Or if it doesn't say ratio, if it says percentage, then you can just do 100% big W, little w. But if it does ratio, the dots is what I'm looking for. Okay. All right. My dog is going crazy. Let me finish this question. Phenotype is 100%. If it says percentage, you can say 100% wire-haired. But since it says ratio, you can do wire-haired to smooth-haired equals to, there are 4 boxes. You can write 4, and then there's 0 of the smooth hair. Or you can reduce it to 1 to 0, whichever one is right, okay? Make sure, okay, I emphasize this here in class, make sure you write out the actual genotype. Sorry, the actual genotype and the actual phenotype. So that, because if you switch, like on the bottom one, you can write smooth versus wire, you can write 0 versus 4. But if you just write 4 to 0, 0 to 4, I don't know which one to switch. Hold on a second. All right, just so this is what he has, this little snake. Okay, let's move on. Clearly the dog is not taking the test. Otherwise, he'd be studying. All right, we're looking at question number 2. Short hair is dominant over long hair. Do this quickly now. This is the first question. Hopefully you got it. All right. You have to get it. If the monohybrid is, if you're struggling with the monohybrid, you got it. You got to figure it out. Come see me ASAP because you're going to be struggling. Okay. But you got this. I believe in you. Okay. All right. Question number 2. Short hair is dominant over long hair in rabbits. 2 heterozygous. So easy, right? Big L, little l, big L, little l. Because they're heterozygous. That's what heterozygous means. Doesn't matter which one is mom, which one is dad. It doesn't even tell you. I don't care, right? So let's fill it out. The box comes down, box goes over, big L, big L. Box comes down, box comes over, big L, little l. Down, over, big L, little l, little l, little l, okay? So what we see over here in terms of genotype, we do have 3 different kinds of genotypes now. I like to write my homozygous in the front. Equals to 1 to 2 to 1 ratio, because we see this one time, we see these ones twice, right? So the big L, little L, you see twice. Little L, I see once. Now, in terms of phenotype, we got to think about which one is dominant, which one is recessive. Long doesn't mean it's dominant, okay? In this case, short is dominant. So big L, it shows the dominant trait, which is short. Big L, little L, it shows the dominant trait, which is short. Big L, little l, it shows the dominant trait, which is short, and then little l shows the recessive trait. Finally, some love for our recessive trait is long. So in terms of phenotypic ratio, it will be short to long. I notice I'm not doing percentage, it's a ratio, equals to 3 to 1. You can also reverse it. You can say long to short equals to 1 to 3. So it's not the numbers that matter. Numbers do matter, but you got to tell me which way, right? If you just read 1 to 3 or 3 to 1, I don't know which one you're talking about. Maybe you got it wrong. If I don't know if you got it, if I can't tell if you got it right, then it's wrong. Okay. All right, let's keep going. Speed round. The ability to curl your tongue up on the sides, the tongue rolling, is dominant. Okay, so big T is tongue rolling. And then not being able to roll your tongue. Whoa. It didn't give us a letter. But you can do this. We got the big T. Well, this has to be, it says dominant 2. That means not being able to roll your tongue. That's your little t. Okay. A woman who can roll her tongue marries a man who cannot. Well, here's the deal. Here's a problem. We got a problem over here, okay? Here's a problem. The man who cannot roll their tongue. Well, not rolling is recessive. So we know the only way to have recessive is to have 2 of them, right? So that's when you have little t, little t. Easy. But now, mom is a little questionable right now. She can roll her tongue. She could be big A, or she could be big A, little a. Well, which one is it? I don't know. I put an emoji right here. I don't have one, but we don't know which one, right? Their first child has his father's phenotype. All right, let's look at the child. Maybe the child would give us some insight. So the child has his father's phenotype, which means the child also cannot roll. Not roll. So if child is not rolled, the child is also little t. Whoops. Use the wrong letter over here. Big T or big T, little t. Okay. Now, well, what does that mean? The child is big T, little t. The child has to receive a little t from dad. Well, where does the other little t come from? Definitely not dad, right? That's not possible. Dad can't give you 2 copies. You get one copy normally. Okay. So this other copy had to have come from mom. How can mom give the child a little t? Well, mom has to be this one. See, it's just a little puzzle. Okay. So mother is big T, little t. Father is little t, child's little t. So these two were easy, but your job is to use the child, figure out what's going on with mom. Okay, cool. So this one's kind of like a test cross, right? We take a dominant and we cross it with a homozygous recessive. All right. Next question about some border collies. Okay, well, the next question is on the next page, so that's kind of annoying. In border collies, the black coat is dominant to red coat. Breeder has a black male as wants and rewards. Okay, so this black coat we know is either big B, or is big B, little b, right? As I'm reading, I want to start writing the words already, or not the letters. The breeder will like to use the dog for breeding if he's purebred. Okay, so this is what we want. And it says, to learn this information, she test crosses him with a female. So it tells you this time we are crossing with a little b. But is it this one? Is it this one? We don't know. But we're going to cross it with little b, because we always do, okay? Even if it doesn't say this, you should know now. We cross with little b. All right, answer the following questions, okay? See the questions. And once again, we have big B, or big B, little b. We don't know. Is it this one? Is it this one? But we're using this to cross with little b. Okay? To see the dogs. Look at the dogs. So cute. All right. So if the black male, right? This is the male, the black dog, is big B, the kind of gamete that it can produce is only big B. Easy. If the red female is little b, the gamete she can only produce is little b. Well, but we know this, right? This is not like an if situation. It is. If the black male is big B, little b, they can produce big B or little b as a gamete, right? You pass on one or the other. Red. Is red a trait we're looking at? Oh, yeah. The recessive is a red. Oh, this is red coat, apparently. Wow. Crazy. Okay. So if the puppies are red, right? What does that mean? Well, if the puppies are red, we know the puppies genotypes are little b. Okay? Now here's the deal. The puppy has to have gotten one little b from mom, because mom can only pass on little b, right? But if a puppy also has another little b, this little b has to have come from dad. Well, can dad be this? No, right? Can dad be this? No, because that dad doesn't have it in little b to give, right? So it has to have come from this. So what does that mean? The father's genotype is really big B, little b, so that the father can give a little b to the child, okay? So imagine this, okay? You want two cartons of chocolate ice cream. Your mom only has chocolate ice cream, and she has two of them. She wants one for herself, she gives you one, okay? Now you have one chocolate ice cream, but you want two chocolate ice cream. Where do you have to get your other chocolate ice cream? It has to come from dad, right? I don't know why that just happened. That's really creepy. Did you see that? Oh my God. Well, anyway, that's kind of scary. I think maybe Zoom is just really happy that I'm talking about chocolate ice cream. Well, anyway, so you got one chocolate ice cream from mom, you have to get the other chocolate ice cream from dad, right? If dad only has vanilla ice cream, two of them, you're not gonna get any chocolate ice cream. In order for you to get a chocolate ice cream and another chocolate ice cream, you have to get one from mom and from dad, right? Dad can't be homozygous. Dad has to be heterozygous with vanilla ice cream to give you the other chocolate ice cream, okay? Hope this makes sense. It doesn't. Well, we got more test cross practice apparently. All right, there's some paragraphs. You can read this on your own. Let's just do the practice. Okay, don't skip it. Okay, if you think you don't understand test crossing, you need to read it, read it. If you need to watch some YouTube, I'm gonna advertise for YouTube. I'm not sponsored, but if you need to watch some YouTube video to get yourself really comfortable with the test cross, do it. Do it. All right, let's look at questions. Phew. We're gonna change color. Okay, it says in fruit flies, red eye is dominant over brown eyes. Let's assign some letters. Let's make red eye big A, brown eye little a, right? Dominant over. So brown is the recessive. You managed to capture a red eye fruit fly. All we know that this fruit fly is showing the dominant trait, right? But what we don't know is the actual genotype. The genotype could be big A, or it could be big A, little a. Which one is it? Again, we don't know. Now we want to determine what it is. Okay, so how do we do this? Well, question A, give the phenotype of the flies. The phenotype of the flies will have to be the red eye. It doesn't say a male. It doesn't say a male. It doesn't matter. Okay, so we take a red eye fly, and then we got to cross it with a white eye. We like the white eye because white eye shows the recessive trait. Wait, no, it's not white eye. I'm losing my mind. It's not white eye, it's brown eye. Okay, because brown eye is the recessive trait, right? The recessive trait is only made possible by homozygous recessive genotype. This is the genotype, that's the phenotype. All right. So we know the brown eye is little a. But we're really like, I'm not going to look at the little a. I see the brown eye. Take the brown eye fly and pair it with the red eye fly. Okay. So then it says, if the two flies have 400 offspring that all turn out to be red eye, circle, underline, highlight, whatever you need to do. Now it tells me to use R. This is so sad. Let me fix myself real quick over here. Dang, we went through this whole way, and then this is why we got to read the questions. Okay, big R, little r. Big R, or big R, little r. Okay, now the brown eye has to be little r. Okay, so it says all the offspring has to be red eye. Okay, so what does that mean? The offspring are all big R something else. Maybe the offspring is big R. Maybe the offspring is big R, little r. But whoa. The offspring cannot be big R because the other parent has to pass on a little r. Okay, doesn't matter. Either way, what you need to remember is that if all the offspring show the dominant phenotype, okay, during a test cross. Excuse me. That means the unknown, right? The unknown dominant one, in this case, is the red eye fruit fly, will have to be big R. Has to be a homozygous dominant. Because if you want all of the offspring, okay, let's take a look. Okay, so this has to be the way, right? We use big R, cross it with a little r, because it's a test cross. This is a test cross part that we pair up on purpose. This is the one that's unknown that we figure out later. Well, how does that work? If all the offspring has to be red eye, when like this one parent only passed on the little r as gamete, right? That's the brown eye allele. But if all the offspring are red eye, that means the other parent cannot give any little r. Like if the other parent also gives a little r, then the offspring could be little r. Anyway, if you're not sure, here's what you do. Just draw two Punnett squares. You draw big B, little b. No. Wrong. Big B, little b. Right? This is one possible test cross. Or the other possible test cross is big B, little b. Little b, baby, little b. Okay? Those are the only two scenarios. If all the children are dominant, you then know the parent is this one. If the children are half and half, then you know the parent is this one. Okay? Cool. All right. What percentage? I think we're just doing this Punnett square. Okay, let's fill out this Punnett square as requested by the question. It's asking me to fill out the Punnett square, big R, little r, big R, little r, big R, little r. Complete the Punnett square. What percentage is heterozygous? 100%. What percentage shows a dominant trait? 100%, because we got to show the big R, right? Big R is dominant. When there's big R and a little r, you show the big R. Moving on. If you perform the same experiment, but this time 200 out of the 400, as in half of them have brown eyes, that means the other half has red eye, right? So that's your half and half situation. Then the genotype of the red eye, that's your unknown one, like the unknown. All right, the red eye, that one. The genotype will then have to be big R, little r, okay? Well, I use B over here. But you get the point. You got this. Okay, we finished it. Page 3 is done. Only 3 more to go. Let's go. Wait, how many pages are there? Okay, so only 3 more to go. Let's go. Okay. All right. Here we go. Apparently you're becoming a milk farmer. In milk. No. It's not milk farmer. It's mink farmer. In mink, the animal. Black fur is dominant to white fur. It doesn't tell me what letter to use. Oh, wait, it does. Whoops. Okay. So we want to say, sometimes I like to write it out. So big B is black. Little b is white. Okay. Just like some of you, I'm not a huge fan of reading, but maybe you are a huge fan of reading, then good for you. Okay. Happy for you. You're already winning life. But if you're not, that's okay. Well, if you don't like reading, what you do is you give yourself shortcuts. What I give for myself as shortcuts is I write something on the side so I don't have to read that paragraph again. I already read it once. I don't want to read it 3 times. That's so annoying, right? Okay, so I'm reading it once. Since the black mink, right, like it's very straightforward. I don't have to read again. Since black mink coat costs more than white mink, I don't really care. You decide to only raise black mink. Well, is big B on its own a mink? No. It has to be big B or big B little b, right? For the black mink. Okay, that's my black minks. Everything is going well, but the guy you bought your mink from seemed a little suspicious. You want to make sure that they are purebred. Okay, you're like, ooh, are they actually purebred? So then you run a test cross. So we don't know if the black minks, right, the ones that I bought was my hard-earned money because I'm a mink farmer, are big B or black big B little b. What I want is this one, but I don't know if that's what I have. Okay, purebred means homozygous, so that's big B. That's what I want, but I don't know if it is because right, all I see is black mink. I don't know which one it is. Hope you get the point, right? Like if I see you having brown eyes, I don't know if you're homozygous or heterozygous. Y'all have brown eyes. That's all I see. Okay, so you do a test cross. The phenotype that we're going to cross is we're crossing black. We're crossing with white because white is certain. We like certainties in life. We also like spontaneous things, adventurous, but having a certain amount of certainty is good, right? Or at least half of it during genetic crossing. So the one that is certain is little b. But I don't see little b. All I see is white. But when I see the white mink, I know it's little b. So I can capture it and cross it with my mink farmer, black mink. Okay, so then what happens? In your first test cross, 30 out of the 60 are black and the rest are white. Dun, dun, dun. Well, what does that mean? Well, it means that I'm having a half and half situation. That means the children that are white minks are little b. Well, how is that possible? This child has to get one of the little b from this parent because that parent can only pass down a little b, right? So this parent will directly pass down a gamete, which is one of the little b's. Well, you got 2 little b's. Where did that other little b come from? It has to have come from the other parent, my unknown. Well, if my other parent is big B, I'm not going to get a little b out of the parent, right? So what that tells me is because there's another little b in the child, that has to have come from my other parent. And if you're not sure, you're going to drop on the square on the side, which we will, okay? But we are sure, okay? And we want to be sure. So you just, if you don't know, write it out. We're writing, okay? So what we see over here is a divide between the dominant phenotype and genotype, ooh, versus the recessive. Why are these circles? Genotype and phenotype, right? So all of those are big B, little b, and they're all black minks as offspring. Like those are the babies. Babies. And then these ones are white minks. Babies. Okay. Okay, so that means my prediction is right. Going from the children back to the parents, like not from parents to children, going from children to parents, we know that the parent is big B, little b. And then I use the Punnett square looking at parents to children to verify that my children to parent method is correct. Okay, cool. Complete the Punnett square. What percentage are heterozygous? Heterozygous are these ones, 50%. What percentage shows the dominant trait? Also these ones, 50%. Done. Next question. Last one on test cross. There were a few test cross questions by accident in the monohybrid, but it's okay. All right, number 3. Think you have a mink? Oh, I'm still talking about the minks. The minks. And there may only be one bad one in the bunch. Oh, those sneaky minks. You decide to do a second test cross on a different mink. So I capture another one of my little animals. I cross it for another test cross. Gosh, I'm really just not tired because you know, gotta catch them all gotta catch all the minks, right? Gotta catch them all. All right, this time, out of the 55 offspring, every single babies, all the babies are black minks. Okay, so if you have all the babies that are black minks, that means the parent has to be homozygous, okay? I'm saying the parent has to be this. The other one is still little b, little b. Little b, little b is test cross. We cross with little b on purpose. Okay. Complete the Punnett square. Let's verify going from parents to offspring. Little b, baby, little b, baby, little b, baby, little b. Indeed. All of my offspring are black minks, okay? What percentage are heterozygous? 100%. What percentage showing dominant trait? 100%. Does it match my question? Yes, hooray. Okay. Done. Well, we only have 2 more types of practice. Let's keep going. We got this. Okay, sex-linked practice. Phew. Sex-linked practice. Sex-linked means instead of our regular big A, little a, little a, little a, we don't have that anymore. What we have is the divide between male versus female. So here's the deal. Males have XX. Sorry, is what happens when you're this tired. Males all have XY. So in your 23 pairs of chromosomes as humans, the last pair of chromosomes that we look at on like one of those chromosomes orders, the males have an XY chromosome. Your X chromosome comes from mom, Y chromosome comes from dad, always, okay? A female, however, has an XX chromosome. So I'm a female. I get an X chromosome from mom and an X chromosome from dad, right? So it's really dad that is determining my femaleness because mom will always give me an X chromosome, right? Like this X chromosome has to be from mom. This X chromosome is also from mom. So the other X chromosome has to be from dad. This Y chromosome is from dad, okay? So why does this matter? Well, we're still looking at traits, so we can still have dominant versus recessive. A dominant allele, or just dominant, you got this, is a dominant allele. I think I should fix it. It just looks so bad. All right. But what does a dominant allele look like, right? Allele is just like how it was before. It's one letter, but it's combined with the X. X big A is a dominant allele. A recessive allele is X little a, okay? Because it's attached to the X chromosome. When we say sex-linked, it's the same thing as X-linked. Like this particular gene is located on the X chromosome. It's not just on any other regular chromosomes. You have 22 pairs of other chromosomes. And they still matter, but they don't directly matter in terms of deciding your biological sex. That's why we say sex-linked or X-linked. It's related to what you're going to be, a boy or a girl biologically. Okay. So now, because this allele, the dominant and recessive, looks the way it is, for a male, you can have either X big AY as a genotype, or you can have X little ay. Those are the two possibilities. For a female, however, you can have X big A, or you can have heterozygous. You can also call this a carrier, or you can have a homozygous recessive. Those are the three, it's getting a little messy. These are the three females, and these are the two types of males in terms of genotypes. But in terms of alleles, you can only have these two alleles, okay? But because male only has one X, he can only be X big AY or X little ay. You have nothing attached to the Y because it's X-linked, okay? Now that you understand this, hopefully, let's answer the question. In a cross between, like earlier I said, in the other crosses, it doesn't matter whether it's a male or a female, in X-linked or sex-linked, it matters. Okay, in a cross between a purebred, okay, purebred means homozygous, red eye, okay? Female, there's so much information here. First off, it's a female. Female, I'm thinking XX. We're gonna put XX right here. And then it says purebred. Oh, it doesn't tell you. Oh, wait, it does. It says red eye is dominant, right? So that means red, in this case, running out of space. Red means X big A is red. That's the dominant allele, okay? That means white eye is X little a. That's your recessive allele. So it says purebred red eye. That means homozygous red eye. X big A and another X big A. It's just like your regular big A, except for you have to attach the XX to represent this is the actual female. And then we're looking at the male offspring. Wait. Oh, then we're looking at the male parent. The male parent is white-eye male. The male part tells you it's XY. The white-eye part tells you it's X little AY. Okay? It's the only way. You can't have a heterozygous male. A male is either dominant or recessive because you only have one shot. All right, let's put the Punnett square together. X big A from mom, X little a from dad, right? The box is still combined this way. So what am I saying? I forgot. Okay. We're combining the boxes the same way. All right, this box, oh, you still want the big A in front of the little a. X big A, X little a. You can call these two, we'll call it later. X big A, Y, and then we have one more, X big A, Y again, okay? So what do these boxes mean? The first box is a female, and it has X big A and an X little a, but the X big A overpowers, the dominant overpowers. Since the female was red eye, female was red eye again. The bottom one is a male was red eye, and a male was red eye as well. But what makes the male red eye, right? Like what makes the male having red eye versus white eye? It's because of mom. Because mom is the one that gives the male the X chromosome. The dad only gives you the Y chromosome. So whatever is attached to the X chromosome that your mom is giving you is what you get, okay? All right, what does the question say? What percentage of the male offspring will have white eyes? So for this question, since it says of the male offspring, it means, try over here, out of all males. Okay, so we're just looking at the male boxes. So in this case, we're looking at, why are these colors are so ugly. Is this, oh, that's just yellow. Okay, we're just looking at the male boxes. So we're just looking at male boxes, just looking at these two boxes. So out of all the males, which ones are, what does it say? White eyes. Well, both of them are not white eyes. It doesn't matter. It's 0%. Okay. A question could say, what percentage of the male has, like, let's say red eyes out of all the males? Then in that case, you would say 100%. But if it says, what percentage of the males are red eyes? out of all the offspring. So that's the case where it'll be 2 divided by 4. So instead of looking at the 2 boxes, in that case, you'll look at, well, that's not a highlighter. In that case, you'll look at all 4 boxes. So the 2 boxes out of the 4 boxes are red eyes, okay? All right. We're on to question number 2. We spent a lot of time on question number 1 because I re-explained everything. Question number 2, we can just do it. Just do it. A female Drosophila of unknown genotype. All right, then that's off to a good start. We're X of some sort. Crosses with a wide-eyed male. X little W Y. So we know X little W means wide eye. It says it right here. X big W is red eye. Okay. Then it says for the next generation. Oh my God, it's a half and half generation. Half of the male and half of the female offspring were red eye, and half the male and half the female offspring were white eyes. What's the genotype of the female fly? Okay. First up, this question will be on the test. But since we're here, let's get through it. Okay. So how do you do this question? Well, the female could either be, we know the female, oh, it really doesn't say. Could be X big W, X big W, X big W, X little W, or X little W, X little W. When in doubt, just draw some Punnett squares, okay? But here's the deal. We can already eliminate one. This one does not work, right? This one, the mom only has X little W. Dad already only has X little W. So you're not going to have any red-eyed child from both parents that are white-eyed. Like, you're not going to have 2 recessive parents and get a dominant child. That doesn't really work, okay? Eliminated, okay? The other 2, let's just draw it out real quick, see what happens. Okay, assuming that the parent is X big W, X big little W, this is the other parent. Okay, draw upon a square. Big W, X little W, X big W. So this is what you want to do on the test. Don't use W. It's real annoying. X little W, X. Yeah, I'm already confused. This is big W. Big W, Y. X, you just got to write your W real big. Okay, so what does that tell us? We're ending up with, let's see. This one is a female red eye. Female red eye as well. Oh, wait. Everybody has red eye. Well, that doesn't work, does it? If mom only has the red eye allele, this is all red, right? If mom only has that, it doesn't work because all the babies will have red. Because first off, the red overpowers the white already, right? So for the female babies, the big W is going to overpower the little W. And then for the male children, whatever mom gives is whatever the child gets, right? If mom only has X big W, you're not going to get any white-eyed male. So this also doesn't work. Okay. All right. How do I eliminate it in a more civilized way? This also doesn't work. This one didn't work. So what you would do, well, we have one option left. X big W, X little W, X little W, Y. X big W, X little W, X little W, X W, X big W, Y. X little W, Y. Okay, so what you see over here is indeed half and half for the females, half and half for the males. Hooray. Page 5. We'll get through this, I promise. All right, question number 3. Okay, all right, this one's easy. It says red eye is dominant and X-linked. X big A. Oh, finally, it's not making me use W. X big A is red. These are the 2 types of alleles. X little a is white eye. Yeah, okay. So we have white eye female. I don't know if it's homozygous or heterozygous, but if it's white eye, that's recessive, it has to be homozygous, right? So it says female. The female tells me it's an XX. The white eye tells me it's X little a, and X little a again. Then we're crossing it with a red-eyed male. A male tells me it's XY. Red eye means dominant, X big A Y. And we fill out the Punnett squares, X big A, X little a, big A, little a, little a, Y, X little a, Y. What's the phenotypic ratio of the offspring? Whoa, here's the deal. When you have X-linked for the children, you need to tell me whether it's a male or a female, okay? So how do I do this? Here's how we do this. This fly. The first box, this is a female that has red eye because the big A overpowers the little a, and big A is red eye. This is another female with red eye. Another male with white eye. Male with white eye. Okay, so your answer should be female. Okay, you can write red-eyed female, right? It says phenotype. So not the A and the little like X and the Y. We're saying the actual physical trait. Red-eyed female to white-eyed male equals to 1 to 1, or you can say 2 to 2. You count the 2 boxes and 2 boxes, right? Done. Okay, last question for X-link. Excuse me with my sniffles. A human female carrier. Okay, we know what a carrier is. Easy stuff. So she's carrier, okay? So we can just write X big X little, and we don't need to keep reading. Well, but we do need to keep reading. The red-green color blindness. So that means X big A is not colorblind. Okay, you can see how I like to write out my alleles. You don't have to write the word alleles. I just want to point out to you what I'm writing out is the alleles. Like, how do I write the letters one at a time, right? But then when you write the actual parent and the children, you need 2 of them. X big A. And then we have, I'm just checking to make sure the microphone is working. You know how sad it would be if the microphone was not working. Oh my goodness. Back to work. Okay. X little a, right? It says sex-linked trait causing color blindness and is recessive. So that means X little a is X little a is colorblind. All right. Then it says we have the female carrier marries a normal male. Okay, because it's a male, it's X and Y. Because it's normal, it's the not colorblind, which is X big A Y. Let's answer the question. X big A, big A, big A, little a, big A Y, and X little a Y. And just to make it really clear, okay, for our last question, we gotta be really good at this. This X big A came from mom. This X little a comes from mom. This X big A comes from mom. This X little a comes from mom. I just want you to know, if you have colorblindness, it's your mom's fault. But it's really your grandparents' fault, which is really your, at some point, colorblindness showed up. But it's okay. We can still enjoy life with colorblindness. What proportion of the male offspring? So because it says of the male offspring, that means we're only looking at the male offspring. The male offsprings are these 2. We don't care what the females are. Oh, not look good. We don't care about the female boxes. We're just looking at these 2 male boxes. Why is it so messy? Okay, so what does that mean? Which ones are going to be colorblind? So I'm going to circle the one that is colorblind. Well, this one is the colorblind one, right? Out of the males, that's just 1/2. 1/2. Really circle my answers. On here, for the answer key so I can send it to you. All right. Okay, we are almost done. Can you imagine going through so many problems? See, if you got every single one of these problems, you're going to get 100% on the test. All right, our last test of the entire year. Let's finish strong. All right. So tired, aren't you? Okay. Dihybrid. We got, oh my gosh, 4 practices. We got this. Okay. Dihybrid. Dihybrid means we're looking at 2 genes. And that means we're looking at 4 alleles. Okay. So you're looking at like big A, big A, little a, sorry, big A, big B type of situation. So let's just go. Let's use this problem, figure it out. Okay, so we have spotted skin, big S, over non-spotted for little s, and woolly hair, woolly mammoth, versus non-woolly, dominant versus recessive. You want to make your big W real dramatic. You're crossing with a heterozygous spotted. So I'm going to write down here. I'm crossing a heterozygous spotted non-woolly, right? So for the heterozygous part, I know heterozygous spotted, right? So big S, little s. Non-woolly is little w. So I know for the second part, it's not heterozygous. It says heterozygous in the front, but it doesn't apply for both of them. The non-woolly means is little w, little w. Next one. This is a man. Marries a heterozygous. Let me just highlight this real quick for you. Heterozygous spotted non-woolly man. It's such an interesting wording. Versus heterozygous woolly non-spotted. Okay. Woolly non-spotted. Okay, see, it's trying to play tricks on us. It's trying to switch the letters. I put S in the front, so I want to put the non-spotted in the front the second time as well. So non-spotted, as you notice, is little s. So I need two little s's. And then it says woolly. Woolly is heterozygous, it says. Heterozygous. So that's big W, little w. Oh my goodness. All right. Now we need a FOIL method, okay? How does one FOIL method? You want to just do one at a time. Let's do the first one first. So as I mentioned earlier, if you watch that part, you want to look at each trait on its own. This is a male, we'll use Greek. Each trait on its own, okay? So first is this one, big S, little w. Outside, big S, little w. You don't want to write as because you need one of each. Inside, little sw. And last, little sw again. Done. Okay, now we gotta do the second one, and I don't have enough space for it. We gotta move this one over a little bit. Okay, now let's do the second one. Second one, we'll use orange. Again, we're looking at one trait at a time, right? So first of each type of letter. So the first of S and first of W. Little s, big W. Outside, little s, little w. Inside, little s, big W. You really gotta differentiate. These are terrible letters. Who did this? I found these questions somewhere. I just copied it. The last is the last of each type of letter. So the second of the S and second of W, little s, little w again. Now we combine them, okay? So for the boxes, I'll show some of the boxes in two different colors so you can see where it came from. You just bring the boxes down. What you don't want is you don't want to do this. Oh, wait. See, I'm already doing it right because I'm so used to it. You don't want to do this, okay? The S's stay together and the W's stay together. Like each trait stays together. But within that trait, the dominant goes before recessive. So what do I mean by that? The big S comes down. The little s follows it. The big W follows that. And little w follows that, right? So between the s's are staying together, the w's stay together. But within the s's, the big goes before the small, just like how we always write it. And then the same thing for your second trait, the dominant goes before recessive, okay? The rest of it, let's just use one color. Okay, big S, little s, right? Big S, little s, big W, little w again. Little s, because we got a little s right here, a little s right here. Big W from here, little w from here. Little s right here, little s right here. Big W, little w. All right, let's keep going. Wait. This one and this one are the same. So let me just copy that real quick. Well, you'll have to copy, right? You might say this is unfair, but I am doing all the talking. You have to do all the talking, so it's all good. But how does one does paste? Oh, there we go. Paste. There we go. Whoa. Okay. All right. Let's write the second row. I get a big S from the top parent, little s from the side parent. Notice that I get one of each copy from each parent, right? One S from one parent, the other S from the other parent. Let's look at W. All we have is little w, no problem. Little w, w. Little w. Okay. Big S, little w, because these two are the same. Little s, little w, little s, little. Okay. Big S, little w, little w, little w. Hopefully you're doing this right. All done. Okay, now on to the interpretation part. The dihybrid is really quite the handful. All right, what do we notice? These, all right, highlighting is fine. These four boxes are the same. I see with my little eye. I spot with my little eyes, however you say it. This two boxes are the same, or four boxes, right? And then we have these four boxes that are the same, double heterozygous. And then we have our last one, maybe it's purple. Oh, that's very similar. Blue. Oh, goodness me. Whatever. This box, and then this last is way too dark. What about this one? Okay, green is not as bad. This box, and then this last box are the same, right? So what does that tell me? It tells me I should use a different color that's darker next time. Okay, our genotype ratio. I got to write out the genotype. Luckily, they're all just the same. All right, so I got this one, I got this one, and then I get this one. And this last one equals 2. 1 to 1 to 1 to 1. Or you can say, because they're four boxes, you can say 4 to 4 to 4 to 4. Doesn't matter. Either way, it's the same thing. All right. Phenotypic ratio. How do we do phenotype? Phenotypic ratio, we're looking at what are the actual trait, right? So the yellow ones, both of these are spotted, and the big W is woolly. Okay, so we have 4 of those, the yellow highlighters. These two are non-spotted, woolly, right? Because woolly is big W. And then we have the green one. We have spotted, non-woolly. And then lastly, we have non-spotted, non-woolly. Okay. We're gonna be really good about this one. We're gonna write out the whole thing, okay? You're gonna be okay. All right. Spotted. Got this. Okay. Spotted, woolly. 2. Non-spotted. It's just really just 4 different combinations. Woolly. 2. Spotted. Non-woolly. The handwriting is really something else though. The non-spotted, non-woolly equals 2. Here we go. 1 to 1 to 1 to 1 ratio. Or you can write 4 to 4 to 4 to 4, and it's done. That's question number 1. We got a few more to go. Question number 2. Okay, we got this. We got this, people. In horses, black is dependent on the dominant gene. Big B is black. Chestnut is recessive. Little b is chestnut. All right, speed round. Okay, here we go. Chestnut. Okay, trotting. Okay, trotting. Trotting. We're doing big T. We're trotting, whatever that means. Trotting gait. Little t is pacing. Oh, it's like how they run or hang out, the bounces. Okay, cool. Let's look at the parents. If a homozygous black pacer, okay, homozygous, it says black pacer. Black is homozygous, big B, big B. Pacer is for T, but it's the recessive one. So that's little t, little t. Crossing with homozygous, chestnut, Potter. What are these words? Chestnut is recessive for B. So little b, trotter, Wait, hold on. Homozygous black pacing. Yeah, we're fine. Trotter, since it says homozygous in the front, it didn't give me any other information. So I'm just gonna assume that it's homozygous for both. So for the trotting, big T, big T, big T. Okay. Well, this one's easy because we get a lot of the same answers. We're gonna be good. We're gonna do the FOIL method. F outside, inside, out. At last. Okay. First, big T, little t. Outside, big T, little t. Well, big T, little t, big T, little t. Easy. Right? Then we have first, little b, little t. Outside, little b, little t. I'm still drawing the bracket so you can see. Little b, little t. Am I saying little b, little t, little b, big T? Last, little b, big T. Then you fill out your Punnett square. And if I give you all 8, you do all 8. 16. All right, let's just sit in silence and get it done. Okay, now we can't sit in silence. How do we fill this out? Once again, the B's go together, the T's go together. If B goes in front of T, all the B's go in front of T. If there are 2 B's, the big B goes in front of little b. Okay, let's do it. Big B in front of little b. Big T in front of little t, but the B's are in front of the T's, okay? And then you just keep going and fine. Okay, so here's the deal. Don't write this on the test, okay? So I'm telling you, I'm warning you, don't do it. Gotta write it out all 18 times, 16 times. Well, or luckily, maybe it's a multiple choice question. It will just ask you what is the appearance of the F1 generation, in which case draw the arrows. I don't care. Okay, the F1 generation is they're all, well, is big B, little b, big T, little t the right answer? No, because it's asking for the actual appearance. So it's really asking me about the physical trait, okay? And the physical trait is big B, little b will give me the physical trait of black, and the physical trait of big T, little t is trotting. I have a lot of these horses. Done. Okay. Last 2 questions. Hope they're easy. All right, number 3. In summer squash. All right, I'm just gonna make the executive. No, we'll stick to it. It's fine. It's W again. Who made these questions? I promise it's not me. In summer squash, white, I like to write it out because it really just helps me not having to look at a paragraph, right? So easy. Little W is yellow. Big letter, capitalized D is disc-shaped fruit, like a circle squash, I guess. And then, oh wait, actually, disc as in like not a circle, like that? I don't know. Little d is sphere. Well, you can see now it's like not that hard. It just takes a little bit of time. Okay. Squash plant is true breeding for white disc shaped fruit. Okay. So true breeding means homozygous. White, big W, big W. Disc shape, also big D, because it just says true breeding. So I'm just going to use homozygous for both. Or yeah, didn't give me any other information. True breeding for, but it's not always the same, right? You might say true breeding sphere, then it will be big W, little d. Right? True breeding for yellow sphere-shaped fruit. I'm gonna write it over here. Yellow is little w, sphere is little d. Actually, in this case, it doesn't even have to tell me true breeding, right? Yellow has to be little w. Sphere has to be little d. Okay, now let's look at the questions. No, we gotta figure out the gametes first. All right. This will give us big W, big D. This one, it's all capitalized all the way, right? You can do FOIL. You should, but it'll give you the same answer. But don't be lazy. Sometimes you really do need to do the FOIL. Otherwise, you might get repeating answer when there's not supposed to be. Right now, it's fine. All you have is the same answer. Wait, see? I'm already doing this wrong. First, outside, inside, last. And they're all little w's. Wow, what are the odds that we have two easy questions in a row? Let's fill it out. Big W, little w. Big D, little d. All the way. All right, this is kind of chaotic. It's better to just do this. Okay. What is the genotype and the phenotypic ratio? Well, if it's just gonna not do the ratio or the percentage, it's fine. I'm the teacher. I do what I want. Okay, so the genotype is 100%, unless I did this question wrong, in which case, call me out. I'll give you candy. 100% genotype will be this. The phenotype will be 100, but if the question says ratio on the test, just do the ratio. 100%. Big W gives you white, right? Why am I not saying yellow? Because it's the big W overpowers the little w. So we're going with the dominant trait. White and then disc-shaped. Wash. We're at the end, oh my gosh, of our hour-and-a-half lesson. How we didn't think we would be here, but now we're here. We are solid for the final exam. These are points on the final exam too. And our test on Tuesday. All right, let's do it. Last one. Let's go. Let's go. Okay. We'll use celebratory pink. In mice, the ability to run is dominant. They're running mice. Okay, fine. Big R for running mice. Recessive are circles. All right, I'm already getting a little confused, so I'm going to write it on the side. Big R for running. Little r for, I'm just going to call it circle. They run in circles. Mice, they run in circles and they're called waltzing. Okay, so these are the waltzing. We have the running and we got the waltzing. Hair color are also inherited. We have big B, which is black hair. We have little b, which is brown hair. Cool, I got my letters. I'm ready to go. They're not X-ing. Let's go. We're crossing a heterozygous running heterozygous black mouse. Okay, I shouldn't be laughing at the running mouses, the mice. Heterozygous means big R, little r. Black mouse, also heterozygous, big B, little b. With a homozygous running, so big R, because big R is for running. Homozygous running, homozygous black mouse again, big B for homozygous black, right? Because big B is homozygous. Okay, so let's take a look. In this case, you should FOIL. FOIL is good. We like FOIL. Let's do it. You have the first type of letter, second type of letter. First of each gives you big R, big B. Outside of the whole thing, big R, little b. Inside, little r, big B. And last, little r, little b. First, outside, inside, last. You notice they're the same. Big R, big B, big R, big B, big R, big B. Are you getting pumped towards the end? Big R, big B. Okay. Now let's do the Punnett square. Here's the thing. We see some repeats. So if it was a multiple choice question, you don't have to fill out all 16, right? Just fill out one row. You know the rest of them are the same. I'm sorry. Yeah, one row. The rest of the columns are the same. Okay, let's fill it out. Big R, big R, big B, big B. Big R, big B, little b. Because we're bring this box down, bring box across. All right, big R, little r, big B, big B. Big R, little r, big B, little b. Down, 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 right? Because these four are the same, they're all repeats. So that's how I'm going to draw it. Again, on the test, do all 16. Parent genotype. It's asking me. I already wrote it out. Big R, little r, big B, little b. Big R, big B, big B. Possible gametes. Let's write it out. From each parent. Okay? Offspring, phenotype, ratio. Notice that it's not asking me about genotype. If it was genotype, then it would be 1 to 1 to 1 to 1 ratio. But no, it's asking you about phenotype, which makes it even better. It's 100%. Do? We got the big R's, right? They're all big R's. The heterozygous still has a big R. So the big R overpowers, they're all running. Nobody is running in circles. And then they all had big B's, right? So that means the big B overpowers a little B, or you have two big B's. Same thing. They're all going to be running black mices. Mice. All right. Last question. You deserve a candy. If you watch the whole hour and a half, you deserve a candy. Let me know. Okay. Homozygous running, homozygous black mouse. Okay. Homozygous running means big R, big R. Black mouse means big B, big B. Why do I got to put all these easy questions for you? Wait, hold on. Oh, wait, we're fine. Heterozygous running. Big R, little r. Now we have a brown mouse, little b, little b. Okay, let's foil. First, outside, inside, last. They're all the same. Big R, big B. They're all the same. Okay. The other one we do on the foil. First of each type of letter, big R, little b, right? Why am I underlining this? I want you to end up with two R's. If you end up with two R's, it's all wrong. You want one R, one B, right? So you got to make sure you get the first of each type of letter. Outside, big R, little b. Inside, little r, little b. Last, little r, little b. And then you put it together. Okay, this one I'm just gonna write the columns because the rows, right, these four are the same on the top. So all of these are the same. Big R, big B, little b. Big R, big B, little b. Big R, big B, little b. Arrow, arrow, arrow. Okay, parent genotypes. We already determined we're just gonna copy it down. Possible gametes, big R, big B. All right, here's the deal. You're watching me do this, right? Feel really confident because I'm doing it. You need to know how to do it, okay? Don't just watch me do it. Oh, well, the offspring running. Oh, they're still black mice. Interesting. Okay, anyway, don't just watch me do it. You need to know how to do it, okay? So don't get into the mindset of, okay, I watched this video, I am good. No, if you did this and you got it right, and then you compare the answers with me, then you're good, right? If you watch me do it, and then on Tuesday when you're doing it, you don't remember how to do it, then you're not good. You just wasted an hour and a half. Well, you didn't really waste it. You got something out of it, uh, but yeah. Well, you've gotten this far. Congratulations. This is the last thing that we're learning that's new for this year. Actually, none of it is new, it's a review. Anyway, it's been a great year. I appreciate you. I appreciate your attention, your effort. You're doing great. Okay. You got this. Your grade is not everything, but your effort, your attitude, the way you treat other people matters. And but obviously how you do on this test also matters. But good luck. I'll see you whenever I see you. Goodbye.